So welcome students. Can you hear me?
Yes, ma'am.
Yeah. So let's begin the main course. As I mentioned, this semester 4 is basically based on Anova, which is known as analysis of variance. Okay? Which is known as analysis of variance. So we call it short form will be always Anova, okay? Analysis of variance. This particular method is a quite useful in the most powerful test, most powerful statistical tools to be used in the test of significance. So when you do the test of significance, you can always go for the analysis of variance. Okay. As I mentioned, we have already discussed a few part of that. This analysis of variance will be morely described or introduced by the Professor R.A. Fisher. Okay. Professor R.A. Fisher. Usually we call R.A. Fisher. That is a Ronald Elmer Fisher. Full form is Ronald Elmer Fisher. In the year of, I will say, 1890 to 1962. Okay, he was like a very great statistician and as well as a professor in the genetic field. Okay. British statistician, but done a lot of research in statistical point of view, and a lot of applications are given maybe in terms of agriculture. So a lot of research papers done using the agricultural research. Another thing, maybe I don't know whether you have studied the mainly we have to discuss here two things. One will be, if I talk about the test, we have a two type of basically test, Z test and T test. Okay. And I think we have already seen the major part of that in the semester 3. So Z test is applied it when when the standard deviation or population variance is known, right? The population variance is known, you can go by the known, then you can go by the Z test, and if it is unknown, then you may go with the T test. And obviously, it is based on the sample size. This test is based on the sample size. If the sample size is large, you can always go to the Z test. If the sample size is small, you may go with the T test. And this is only to comparing the two population. When we have a two groups, right? When we have a two groups, let's say group 1 and group 2, or maybe two classes, let's say two classes are there, like the classes of SYBSE of Junjunwala College and the classes of SYBSE of let's say KC College. And if we're comparing their performance in terms of statistics, mathematics, and other activity, so you may always go for using either this or this based on the criteria which is given to us. You want to compare two types of a brand like Coca-Cola and Pepsi, like Coca-Cola and Pepsi. So okay, two types of brand. So you can just look at these two concepts. So when you just want to compare the two groups, we always go by either Z test or T test based on the data, whether the population variance known or unknown. Okay. But just imagine if we have a three classes, if we have a 3 classes, let's say FIBSC, SYBSC of our college, AC college, and let's say Ruya college. Okay, then can I go for a testing? Can I go for the testing of their performance in terms of marks? So in that scenario, we have to go by the analysis of variance. We can't use this Z test and T test. So Z test and T test is only applied it when we have two group of populations. Okay, then you can always go for Z test and T test. But once it is three or more group, when it is three or more group, then we go by, so three or more groups, when there we go for, then we go for ANOVA. Okay, but we go for ANOVA, but make sure that before going ANOVA there are certain assumptions. There are certain assumptions, and that assumption has to be satisfied, then only we go for ANOVA. But actually you should know now, like, I cannot use T test, I cannot use Z test, because of I have a three or more population group of populations. Okay, so but I can use ANOVA now, but how to use it and what are the conditions to we need to be satisfied, and then further I can use it. Okay, so making the assumptions of ANOVA, okay, keep that all assumptions satisfying. Assuming that it is satisfying, one may go by the, one can use it ANOVA. So this is some of the examples where person can be used. So but if I talk about the agricultural example, if I talk about the agriculture example, there are very famous example. Maybe sometime we have discussed that example. Like when you have a certain plot, right? Everybody do love farming, right? Everybody do the farming. Suppose I have this a particular plot, okay, and let's say I have a this some portion. Let's say this is my. I have divided this portion. Maybe you can assume it is equally distributed. Just now you can just assume it is equally distributed, but it is not. But just assume that I have a like equal amount of equal area. Okay, I will just assume it is equal area, equal size and shape. Okay, and so see, I'm just putting some condition. So if I'm using the agriculture example, okay, so agricultural examples, you may understand how this ANOVA can be applied. So I have this plot, okay, in maybe in your village or in your area you have a plot, and what I did, I have divided this area particular plot into the 4 section, assuming that having same size and same width, means the area is same. Okay, now what I'm trying to do here, suppose I'm just doing the wheat plotting here, okay, or rice plot. I just want to uh, make a rice, okay, rice ka plotting karna hai. So what we need to do here, if I am using the condition of, let's say, suppose I am taking the fertilizers. Okay, so I'm just trying to do the putting some fertilizers. Fertilizers, you everybody know what is a fertilizer. Okay, some kind of it is a mixture of all uh, protein, I can say. It's like a mixture of all stuff. Okay, one can say like nitrogen, potassium, nitrogen, etc., etc. Okay, so that is all basically. So we call it a fertilizer. So let's call the fertilizer. So I have fertilizer A, fertilizer B, fertilizer C, and fertilizer D. Okay. So I have applied it these all fertilizer to this particular plot. Similarly, all plots. Okay. Then whatever output I will be getting from A, B, C, D, that is called yield. Okay. So what is the first step? I have a plot. तो उस प्लॉट को मैंने इक्वल शेप एंड साइज में मैंने डिस्ट्रीब्यूट किया. फर्दर आई हैव अप्लाइड इट डिफरेंट fertilizer. Okay, maybe that fertilizer may be potassium, nitrogen, etc. I have applied it, and the things which I got it from this particular plot, that's called yield. Now yield means it can be type of wheat, it can be type of rice, what amount of rice I will be getting. So let's call it like from A I will be get let's say 100 quintals of rice, 100 quintals of wheat, like that. Okay, so that is basically called as the yield. That is called basically yield in terms of agriculture I'm talking about. Okay, but if I talk about in terms of the classes of performance, I will be get the marks. So marks will be my yield. Just try to understand these some of the statistical term, because maybe when you just solving a different type of problem, you may not come across the same meaning. Like if I talk about the brand, okay. So if I say Coca-Cola, so what amount of like Coca-Cola you drink? So drinking amount will be the consider, like 5 ml, 10 ml, 20 ml. Okay. So how much amount of Coca-Cola you drink? So they will say like 20 ml we drink, 30 ml we drink. So that is called basically yield. So whatever things we will get it from that particular category, I will be say experiment, that is called basically yield. So and then now whether, so once you get the yield of that particular plot, like from ABCD, you may get the maybe yield in terms of some number. Okay, and that number will shows a lot of information. So this is my, from A I will get some yield, from B I will be get some yield, from C, from D. Now I am interested to understand whether these, the yield which we got it, which fertilizer is better. Like if I'm saying that after doing everything I got like fertilizer B will be the giving the more amount of yield, means it's giving the like more number of quintals of rice to me. Obvious, I will go by the fertilizer B next time. I'm not going to use fertilizer ACD. Getting my point? So how do you decide? That is the question arises. Okay? So for that purpose, we need to go by Anova. Okay, we need to go by Anova. So understand this concepts, how this particular Anova is useful. Okay, Anova is quite useful. Even maybe when you are going to do the projects, this technique you can use it. Like suppose you want to compare the effect of let's say effect of COVID-19 on the patient. So let's say I'm considering the patient age. Let's say age will be starting from 15 to let's say 20, or let's say 10 to 20, 20 to 40, 20 to 30, 30 to 40. So I'm just comparing these three groups. Say three groups में कितने patient हैं? Just find out. And you can compare, the more effect will be of the COVID-19 will be in which group. Okay, so you can test the Anova. It means you can perform Anova, you can work on Anova, you can just tell whether this particular things will be more effective. Okay, so this is the conceptual knowledge of the Anova which you have to use. So what is the basically, this is the Anova. What actually the aim of Anova is that you need to, as I mentioned, it is analysis of variance. What is the full form? Analysis of variance. So variance means that we need to understand their variation. Their variation. So variation means what? So the variation means obvious when you do the plotting and everything, when you do the farming, it is not exactly what you are expecting. When you just used here fertilizer A, you thought no, there should be the 150 like a quintal of my rice. That is your expectation. That is your expectation, right? So, so the variation is something like that, which is split into the two part, and this is called as assignable variation, assignable variation, or sometimes it is called causes or assignable causes or variation or causes. Okay, and there is a chance causes or chance variation or causes. Okay, so the variation means your total variance is split into the two parts. One will be assignable variation or assignable causes. Second will be chance causes or chance variation. Now, assignable variation means what? Which is in your control. Which is in your control, right? This is in your control. And chance variation means which is out of control. Means it is not in your not in your control. Means we can't do it. This is not possible to us. Like it, like it can be happen. It can be happen. In unavoidable. Like maybe something happen, and because of that you cannot do it. Okay. So for that purpose, like you cannot work on that. But assignable causes can be reduced, can be manageable, that you can work. For example, suppose you have a plant tree. Okay, suppose you have some kind of tree, and you know that in that tree, basically we need maybe the every day we need maybe that 20-liter water. 20 liters water we required in means per day we require. Okay, so that is that is in your control. But what happen if I'm reducing these 20 liters by 10 liters? Can it be it give the same amount of mango? Can it be give the same amount of leaves and etc etc? No, it's not possible. It's not possible. So what I'm trying to say, if you may not give the sufficient things to particular let's say tree or maybe plot or maybe like timing to the studying purpose, then obvious the whole thing converted to the variation. It may be the part of luck only. It may be the part of luck only. So your entire things will be converted into the variation. The very important the things that we need to be reduce this. Obvious, we need to be reduce this chance variation. Obvious, na? Because error. This is nothing. This is also called as error, or it is called as error. So error we try to reduce. Error we try to reduce. The overall aim of ANOVA is that you need to control this and try to minimize this. If there will be no error, that means obvious like the model will be the good fit. My model is good fit. Okay? So I can go ahead with the model. So this is the one concept which you can always take care. Then there are so many things which you can go ahead with this. So you can do like you can do the separation of this ANOVA. Means whatever the like fertilizer ABCD you have, you can separate the variation for due to the assignable variances and due to the chance variation. So you can separate their sum of square, you can separate their variance, and you can check whether that particular variation is effective or not. Okay, that is all we will do slowly. But before I'll do that, we have a certain assumptions which you have to be understand. And before you going with this performing ANOVA, you must be check this all three assumptions. Okay, three assumptions. So that I'm dealing with now. So what is the assumption? Assumptions of Anova. Okay. So the first assumption, you need to be understand these assumptions. The observations are independent. The observations are independent. I hope you can understand what is the meaning of the first assumption. See, observations are independent means what? How do you understand? What do you understand by these observations are independent? So there are several techniques. There are several techniques to check whether the observations are independent. We assume, na? Let x1, x2, xn are independent random sample. We always assume, right? You always assume that they are independent. So as I mentioned, if I have applied it fertilizer A here, fertilizer B here, they are independent, right? Somewhere I'm doing like I have just given students to do this task and it is same task, but the thing is that they don't know each other, like examination. So if you do the offline examination, you sit together and write the paper. You sit like you are sitting all together, but you are writing the paper by independently. You're not taking. I hope everybody is doing the same. So they are. Everybody is like it's not like you're taking the help of your friends and you're writing the paper. Right? So you're appearing. You're writing your own paper like independently. So that's why it is called as all observations are independent. Like your blood group and your neighborhood blood group, that is independent. So these are some examples where the we can call these observations are independent. So mainly this condition will be satisfied, and this is the way we can always check whether the observations are independent or not, and there is no any other way to be checked, but other assumption we can try to check it. And one second assumption, the parent population from which think just a minute, just so parent population from which observation observations are taken is normal. Means somewhere they're talking about the concepts of normal distribution. I hope everybody knows now what is a normal distribution. Okay, so we will not going to discuss much about the normal distribution, but briefly I will tell you. To understand the normal distribution in descriptive statistics, I think I only taught you several technique to be check whether the data which we have and which is coming from the parent population, whether it is normal or not. First example will be stem and leaf diagram. Second will be quartile plot is not there, but generally you can call it as it is a box whisker plot, right? Third technique, histogram, right? So these are the few I can say diagrammatic presentation of the data using, one can check whether the parent population is really, from parent population, the data which we have selected is really follow normal or not. Okay. So this is the second assumptions. And the third assumption which we need to take properly is various various treatment and environmental effect are additive in nature. Additive in nature. So what is the meaning of third assumption? See, the third assumption, the treatment which I'm considering, understand the last point, last word. It should be additive in nature. Effects, effects, the treatment effects, various treatment effects. So here, if you just look at this plot, I have a four treatment. One, A, B, C, D. So fertilizer A is my treatment. So that treatment I'm giving to this particular plot. Similarly here, similarly here. So these all treatment effect should be in additive in nature. Should be in additive nature means something like we have something plus A plus something plus B something plus C and so on. Okay. It should not be multiplicative in nature. Do you remember this is same thing if you remember we have seen in time series.
Yes, sir.
Yeah. So in time series we have see additive and multiplicative and mixture both. Okay. So in certain cases like addition is only possible, not multiplication. So if the multiplication is there, we cannot use ANOVA. We have to use some other technique which is called ANCOVA something, analysis of covariance, because there is a intra relationship between A and B, because we assume that A and B are independent, right? We have just seen the properties like these observations are independent. So that is the very very important. And this environmental effect means this is just make the effect like maybe which is not in control, maybe which is in control, but that is also should be under the control, right? It should be additive in nature. Okay, so checking this, I will say point number this and this is a manually it is not possible. Logically, if you just explore it more about this, you can understand. But second, definitely you can work second. So mainly we will tell you that you can go by the observation which is coming from the parent population that is assumed to be the normal, and these only we can check only using the statistical tools, which is not possible to do the manual checking. Manual means you cannot do the offline checking. So when you can use some of softwares like R software, SPSS software, Excel software, okay, then only you can work on that and you can check whether they are normal or not. Otherwise, manually it is not feasible to check the concepts of independence and normal. Okay, then this is the one part. So other thing will be like we have an uh, theorem which I just want to discuss, which is called as Pokhran theorem. Okay. Pokhran theorem. So what Pokhran theorem says, so whole properties like whole Anova will be using this theorem. Based on this theorem, we are assuming that this theorem and based on that we are proceeding with Anova. What it says, let x1, x2, so on, xn denote random sample, denote random sample from from normal distribution, from normal population, I will say normal population with mean 0 and variance sigma square, and let the sum of squares, sum of squares of these values, sum of squares of these values can be written as, can be written as summation xi square is equal to q1 plus q2 plus on qk, where qi is a quadratic form, quadratic form of x1 comma x2 so on xn with with degree of freedom. Df means degree of freedom rj, and j takes value 1, 2 up to k. Then the random variable, random variables q1 comma q2 so on qk are mutually independent and qi upon sigma square is a chi-square variate with rj degree of freedom if and only if, so we can use I double F. Okay, if and only if means it's a both conditions, reverse and converse both. If and only if summation of rj is equal to n. Okay, so notationally I will be say qi upon sigma square follows chi-square with rj degree of freedom. Putting this condition, if and only if condition, summation of rj is equal to n. So this is the Poporan theorem, which you have to be just go through the basic conceptual and understanding. I will be telling within one step. So first of all, what is this quadratic form? What is this quadratic form? Quadratic form is something like, I will be say x dash qx. Okay. Quadratic form means it is something like that, x dash qx. This is called quadratic form. Okay, this is called as a quadratic form. And if this divided by sigma square follows chi square with whatever respective degree of freedom, means I will assuming Rj here, but putting this condition, sum of Rj should be equal to the number of observation, then the Cochrane theorem satisfy all the condition and you can perform Anova. Means that is a one theorem which you can just understand how it works, that we will see in the next class, that we can go through that. But the only thing is that you need to just understand these observations coming from the normal populations, and the sum of square can be written as like this. Sum of square, everybody knows what is sum of square. Means adding that everything you can get it as sum of square. Summation of x square is the sum of square. And this qi is a quadratic form which having some degree of freedom. Okay, so we will discuss more detail what is the degree of freedom. And then obvious these qi's are mutually independent, and the qi upon sigma square means this is the quadratic form. Qi is a quadratic form, divided by sigma square follows chi square degree. Okay. And then putting this condition Rj is equal to n, if satisfies, then one may satisfies this Cochrane theorem, and one can easy to apply the concepts of the Anova. Means one can perform Anova. Means I will directly. This is I hope I have covered the basic concepts of Anova. What is Anova? When do you apply? Who is invented? Where it is used? When it is used? And how to apply? That is all theoretical things we can see in the coming session. And main aim of Anova will be just doing the separations of variation using assignable variation and using chance variation. Okay. So main motto of Anova is this only. We have to separate the assignable chances. We have to separate the chance variation. Okay. Then how to separate and what is the procedure, that we will cover in the next classes. Okay. And if you have any question, you can discuss, no problem. So I have already sent a mail to everybody. I think few of them is already accepted. Okay, so you can check your mail. Now oftenly you can check your mail, because it is like very important to very important, means professionally I will say very important, because you may come across more things